All right, so let me go ahead and try to start this walkthrough of downloading um, sequences from the NCBI database and to get them into some format that we can upload into Mega, and how to align and then run the sequence. I mean, there are going to be a lot of steps here. You can stop and rewind. I'll try to walk through and explain things as I go along. Okay. So the first thing I want to do is to go to the NCBI database. NCBI database, I have it there. So it's a National Center for Biotechnology Information. That's not where I wanted to go. NCBI database. Okay. And when we get there, we're going to be running a search. I've got it set to nucleotides. So if you click on nucleotide, it'll tell you that you're searching for nucleotide sequences. I did this because I wanted, most of the information that's there, I think, are in nucleotide sequences. There are a few sequences that are there as far as the entire collection that go in different directions. Like you can get whole genomes, you can get protein sequences, the amino acid sequences of proteins. Let's just keep things relatively uniform, and because it's a pretty broad net, we can all use nucleotide-based sequence searches for this exercise. Okay. And so what I'm going to do here is to just type in the name of an animal that I've chosen. I'm looking outside my window, and I've got a little fence lizard crawling up and down on the wall outside, and I call him Doug. Okay, Doug is the name of whatever fence lizard is crawling around outside my garden. And so Doug's species is Scaloporus occidentalis. Okay, so Scaloporus. I know I could have selected the suggestion that came down below. I could have done that, but I don't necessarily want to search for mitochondrion, so I'm just going to do a general search based on my species. Okay? I don't want to limit the scope of my search, so I'm just going to go with species. This way, if there are any sequences that are available for my species, I'll get to choose. I won't necessarily have to look only at mitochondrial sequences. Right? So click on the search button, and Scaloporus occidentalis voucher. Then that's the voucher is the exact sample. This gene is NADH dehydrogenase subunit 1, or the ND1 gene, and it's a complete sequence, and it's mitochondrial. Looks like most of the things that are here are the same thing. I'm going to go ahead and select that. Okay. So when I click on that, I actually get to the complete record. And so this tells you about where the sequence information came from. You get a little taxonomic information, the publication, the authors. This is on a paper called Phenotypic Evolution in High Elevation Populations of Western Fence Lizard in the Sierra Nevada Mountains. So these are probably lizards that were collected up in the Sierra. And when you get down to the bottom, this is the actual sequence that we're looking at. This is what we're going to be comparing between different species, right? All the A's, T's, C's, and G's, they correspond to the sequences of NADH dehydrogenase subunit 1 for this particular lizard that was sampled and sequenced. Okay, and so that's just one record. Now, okay, you can play around with this. You can try to get a sequence that you like. And what we're going to do is, this is going to be the basis of our search for other similar sequences, right? I know I want Scaloporus occidentalis in my sample species. Let's see if I can come up with a group of organisms that are kind of closely related, that have similar comparable sequences to Scaloporus occidentalis. And so for this, you run BLAST. Okay, so BLAST is, as it says right there, find regions of similarity between this sequence and other sequences running BLAST. This is basically a lot like a Google search, only in this case we're searching for sequences that are similar to the A's, T's, C's, and G's that are there in my samples. So I can run for, you probably don't want to touch anything here. You might need to make adjustments based on whether you need highly similar sequences, you want to get close to related sequences. If it turns out that all of your sequences are looking like the same species, you might want to broaden your net. Let's go ahead and broaden the net a little bit and look for slightly more similar sequences than just the just things that match up perfectly or nearly perfectly with the sample that that I'm putting in. Okay, so then you go down here and click on BLAST. And this is a part that takes a little bit of time, depending on how fast your computer is, how fast your connection speed is. It tells you how often it's going to be updated. Okay? And so this is this can sometimes last a few minutes. Again, it depends on how much data there are in the system and whether you choose highly similar sequences or more dissimilar sequences. I think because I chose more dissimilar sequences, it's going to be taking a little bit longer. But that wasn't too bad. Okay, so now it's done, right? And so what it's done is it's taken my sequence, this is the one I had up here, and it's looked for all the sequences that are comparable, okay? And so, yeah, it looks like the first, many sequences that are up here are actually the same species. And so if I were to use, I'm going to unselect all these, right? And so if I were to use this one and this one, that would not give me a good analysis because I'm actually using two samples of individuals from the same species. The point of this exercise is to generate a phylogenetic tree, a reconstruction of the phylogeny of species that are different species. And so I really only need one sample of Scoloporus occidentalis, right? And so I can scroll down. These are all Scoloporus occidentalis. Now I've got another species here, Scoloporus cautus, okay? It's also NADH dehydrogenase subunit 1. It's also complete sequence. And so this looks like a good match for the gene that I'm sequencing, that I'm looking at up here, Scoloporus occidentalis. And it can scroll down further. Here's another species, Scoloporus, another fence lizard, Scoloporus cowlesi. Okay? And it also is a NADH dehydrogenase gene, complete CDS mitochondrial gene. And here's another one, Scoloporus undulatus. But look, this is a, the description is different, right? So if I were to look at what it says here, go to alignment, Scoloporus undulatus, it does have the NADH dehydrogenase subunit 1 gene, complete CDS, in it. And so I think it's okay for me to use that sequence. We'll find out. I mean, if it turns out that it's not looking good, I can go ahead and eliminate that sequence in the editing, right? So I've got Scoloporus undulatus. Here's another one, Scoloporus tristicus. It's also NADH dehydrogenase subunit 1. And cowls, I already got that. Undulatus, cowls, undulatus, tristicus, cowls, undulatus. Here's another species, Scoloporus virgatus, right? And so, again, looking at this, it says complete CDS, mitochondrial gene. I think this one is going to be comparable as well, right? And so now that I've got, looks like I've got 6 selected sequences for Scoloporus occidentalis and several other species of Scoloporus. We're going to download a FASTA file. And I tried this before, okay? So if you go to download, you'll have the option to download as a FASTA. If you do that, it doesn't work, okay? So don't do that, okay? So when you want to download your FastSave file for use in Mega, you'll need to go back to GenBank. You've got your sequence selected. Now you go back to GenBank. It's a page similar to what we saw before. And I've got the 6 items. I've got Scalopris occidentalis, Scalopris cautus, cowl's eye, undulatus, tristicus, and Scalopris virgatus. Okay? And now that I've selected them all, I'm going to go ahead and send to File. Right? And for the format, I'm going to choose FASTA. Okay? You can say FASTA. Technically, it should be FASTA, right? So I'm going to choose FASTA, and then I'm going to go ahead and click on Create File. And now you saw that. The sequence that I've downloaded, it's in my Downloads folder. It's called Sequence FASTA. .FASTA. FASTA is what comes after the dot. I forget what it's called. And so now that I've got my sequence downloaded, now I can go ahead and go to Mega, right? And so for the Mega program, you'll have downloaded it into your Downloads folder. I'm going to go ahead and open up MEGAX. Okay, MEGAX is the version that we have now. And now that I'm in MEGAX, this is the interface window. And I'm going to click on Data, Open File Session, and I'm going to look at my Downloads folder and Sequence FASTA. Right? And so if I open that, it asks you first whether you want to align or to analyze. We are in the process of needing to align. Right? So I click on Align, and there are my sequences. I'll scooch this over so you can see the entire window. And so yeah, there's a scroll bar. And so the entire sequence goes on for quite a ways. And remember Scalopris undulatus, that's one that had more information than just the NADH dehydrogenase gene. There's a lot more information on that one sequence I don't have with the other ones, right? And so what I'm going to do is I need to align this, and then I'll be editing out some of the sequences that don't match up, right? In fact, you can actually look at this. This is color coded, and so see how all the G's and T's are lining up for all the other sequences except for Scalopris undulatus. Well, that's because Scalopris undulatus is not aligned properly with all the other ones. Okay, so I'm going to go ahead and align it. But the first thing I want to do is to get rid of all the extra information here. I'm going to double click on this. I'm just going to put a shorter version of the name. You'll be happy you did this later on. So Scalopris occidentalis is the first record, and so I'll be abbreviating that S. Occidentalis, Scalopris cautus, S. Oops. Cautus. Next one, S. Cowl's eye. Next, Scalopris undulatus. S. Tristicus. Oops. S. Virgatus. Okay, which is going to make our lives a whole lot easier. Okay. And now what we're going to do is align the sequences. Now, you can't see this, it's outside of the window, but up on the very top, the menu bar, if I click on alignment, you can see the drop-down menu coming down. I'm going to go ahead and align this by ClusLW. Okay. ClusLW is one alignment protocol. I can align by muscle. Both of those work. I'm just going to go ahead and say align by ClusL. I forgot to select the sequence that I want to align. Select all. Yes, I'm going to go ahead and OK. Right. And for some reason, this graphical user interface is not showing me the choice that I have at the bottom. As far as the options go, we're not going to get too fancy. We're just going to play around with use of default settings. But I know that in this area right here is the go ahead button. And so that's the OK button. So I actually clicked on that. And now it has done the alignments. And when I look at the sequence now, using the scroll bar at the bottom, okay, I can see that, yeah, it's the, for the portion of the sequences that actually match up, I have things really nicely aligned. Okay, so if I go to the beginning of this part, remember how all these guys were lining up for all the other species, but not for Scalopus undulatus? Well, I've taken care of that. Okay, I've taken care of that entirely. And so now what I can do is I can go ahead and cut out the parts of the alignment that don't matter. Okay, we don't care about all the bases to the left of the matching sequences and all the bases to the right. I can go ahead and get rid of them. Right? And so I'm going to select one corner of this. I can scroll all the way to the beginning and use the Shift button. I've selected that entire block, and I can cut out that block of sequence, right? Okay. It's all stuff that is information that we have for undulatus, but we don't have information for the other species, so we just want to get rid of that. Okay. And so now the sequence begins with all these sequences that match up, all the A's, T's, G's, T's, right? And I want to do the same at the other end, right? So at the other end of the sequence, I probably have more information from undulatus than I do for the other species. There you go, right? And so I can go ahead and click one corner of the block that doesn't have any meaning for us. Click on the other corner, and you hold the Shift button down, click on the other corner and cut that out. Okay. And now I'm down to just those bases in which I've got matching information for my 6 species, right? So this is a nicely edited and aligned data set. I'm going to go ahead and save this. Save. So I'm using the drop-down menu. It's Save Session. So it's basically the same as Control S. And I'll call this, leave it in my, notice that it is in MAS with the MAS suffix. It's an alignment session, and it's a mega, I think it stands for mega alignment session. I'm going to call this Scalopus. The downloads. Okay. So there you go. And now this sequence of completely aligned information is there in my folder. Now I can go back to the other window. You can see the other window over here. Right? So click on the other window, bring that up. Now I can actually open this up in Mega. So I'm going to open up a file session, and here's the Scalopus. Oh, you know what? I forgot to do something. Never mind. Okay, so back over here, I want to go to the, I want to go to the Data drop-down menu, and right here it says Export Alignment. You can probably see just the very right-hand side of that. And I'm going to choose Mega Format. Okay. And now it's going to be saving my file as Scalopus Meg in the Downloads folder. Click on Save, and there it is. And now if I go to Mega, open up the data file, open up a session, the Mega file is there. So scalopus.meg is the file that I want to open. Okay. And so if you want to see the data, that's what the data look like. It looks a little bit different here because what it's doing is it's taking the foundation sequence and saying, well, here are the differences from the very first sequence. So the first sequence, the reference sequence is Occidentalis. So this is just telling us that, well, Occidentalis for this base right here, for that A where you've got all the C's underneath it, Scalopris occidentalis has an A there, everybody else has a C there, right? And whereas in the base over here, Scalopris occidentalis has a C in the base just before that, and everybody else has the same. That's what the dots stand for. Okay, so that's how to read this. But we don't really need to look at that. What we're going to do here, okay, so if you've gotten this far, you've gotten your sequences downloaded in a FASTA format. You've gotten your sequences aligned using Mega. Okay. You imported it using the alignment function in Mega, and you're able to truncate the names, so you only have the names of the species without all that other extraneous information. You have aligned it, and then you've clipped off the portions of the data file that don't really match up. Okay. And now your work is basically almost done because Mega does the rest for you without much thinking. And so the two things I want you to do is to, one, calculate the genetic distance matrix. And you can do that by clicking on distance, compute pairwise distances. Would you like to use a currently active data file? Yes, I absolutely would. Click on that. And we are going to just use the default settings. Boom. There you have it. Okay, so this is a genetic distance matrix. It's a pairwise distance matrix, which basically allows you to compare sequences. All those, nearly 1,000 bases worth of A's, T's, C's, and G's. Well, you can read that by saying you can tell approximately what proportion of those bases are different between Scalopris occidentalis and Scalopris cautus, right? So species 1 is Scalopris occidentalis. That's up here on the 1, right? Scalopris cautus, where column 1 matches up with column 2. This corresponds to the proportion, the percentage of bases that are different between Scalopris occidentalis, that one sample we had, and Scalopris cautus, right? There's no data for where 1 crosses with 1 because that Scalopris occidentalis compared with itself. It's obviously going to be the same, right? So if you wanted to compare Scalopris cowlesi with Scalopris tristicus, you would have to go to column 3, that's Scalopris cowlesi, and then drop down to Scalopris tristicus. And here we've got a 0.0207 difference, less than 1% differences. That means Scalopris tristicus and Scalopris cowlesi are very similar in their sequences, right? And so that's how you interpret the pairwise distance matrix, right? And so when you get to this point, you will probably want to use, if you're on a Mac, Command Shift 4, okay, Command Shift 4, and that will give you a different icon, and you could select your pairwise distance matrix, and this is going to be something that you will be incorporating into your presentation, right? And so that's one item. The other item that you'll want to have for your presentation is the cladogram, the phylogenetic tree. And again, you don't have to think about this too much. The one thing, you could play around with these others. I'm going to ask everybody to use the construct or test the UPGMA tree, which is actually using the same protocols that were there in the Clustel W alignment protocol. So to keep things consistent, we're going to be using the UPGMA tree protocol. And would you like to use a currently active data file? Yes, I certainly would. And again, just go ahead and use the default settings, and you get a tree. Okay, the tree pops up. Look at that; it's beautiful. Right, and you can make it bigger. If you don't get these, if you do this and you don't get the numbers on the branch lengths, these branch length numbers are really kind of important. I want you to have them, right? And so if you don't have them, you might have to come down to this thing right here where it says there's a little icon here with a 1.1 on it. If you click on that, it disappears. If you click on that again, the branch links come back. Okay, so the last time I used Mega, I had the branch links there, so it saved my my preferences. You might not have the branch links showing as part of your preferences, right? And so if you need to click on that, do so, because when you collect this tree by using Command Shift 4 or whatever it is in Windows, you can select this, save the image, and now you've got an image of your tree to incorporate into your presentation. Okay. Now, isn't that easy? Okay. Yeah, there are a lot of steps, and hypothetically, you can be doing this whole thing ahead of time on your own. If you run into any snags, come and you bring them to class. Come and talk to me. There is one thing that I want to share with you, though. Okay, and it goes like this. Okay, so back here on the Alignment Explorer, remember how I told you that I would like you to incorporate some type of outgroup, right? Right now, I don't have an outgroup. These are all part of the genus Scoloporus. And so if I wanted to make a really good tree, right? If I wanted to do that, take that one extra step that would make my tree a little bit more professional, I want to incorporate one more sequence, or at least one more sequence, in which I've got a closely related species that is not part of the clade, right? So Scoloporus is a little lizard. It's in the iguanidae, so I can look for iguana, the great green lizard, great green iguana that we find in tropical forests. But what I'm going to do here is I'll be using the, I think it's data. Okay, and under the Edit drop-down menu, I can have Insert Sequence from File. And again, you're probably seeing the ERT sequence from File, but if you see the entire drop-down menu, it would say Insert Sequence from File, right? Okay, so I'm going to do that, but I need to actually get the file first. Okay, so I'm going to come back to my browser window, and I'm going to do another nucleotide search. This time, I'm going to choose another species of lizard. Okay, and so yeah, I know a few things about lizards locally. So the next most common iguana lizard in this area is called the side blotch lizard, and its scientific name is Uta. Right, so you do need to use the Uta, the scientific names for the species that you are searching for, because common names are probably not going to be there, right? Because people publish in all different languages, and so I'm not sure why I'm getting all these Pongo, which is the orangutan, right? But I'll be looking for Uta. Maybe I'll go ahead. Stans Brianna. I'm just going to go for NADH dehydrogenase, because that's the enzyme, that's the gene that we had for the Scoloporus data. I'll go ahead and put 1 there, so I'll make sure I'll get subunit 1, click on search, and here we go. Okay, Uta stansburiana NADH dehydrogenase. This looks like a perfect thing for me to use. I can go ahead and just click on that one. That's my outgroup and sequence. And go ahead and send that to a file format, FASTA, create file, and now I'm getting another sequence. So the sequence 1.fasta, that's the one that's got my data for the Uta, the outgroup. All right. So now I'm going to go back to Mega. Open up my Mega window again, and go back to data. No, it's edit. Insert sequence from file. Okay. And now I can go ahead and add the Uta stansburiana. That's sequence 1. Ignore. Okay. And now the Uta sequence is right there on the top, right? And so I want to change the name on the front to UTA. Let's go ahead and tell me that's my outgroup. Okay, so now we know it's the outgroup. So in case you forget what your species are, because probably most of you are not terribly familiar with the species that you are going to be analyzing. And now that I've got my 6 Scaloperas and my 1 outgroup, I need to align again. Okay, so I'm going to go ahead and use the, nope, align protein doesn't work. I need to go to the alignment menu, drop-down menu from the top, align by clustale. Go ahead and select all and run the clustale program. And now things are aligned. And now now I need to export. I can save the session. Saving the session is a good thing. Maybe make this one different. Call this one .uta. UTA. All right, and I'm going to export the alignment in mega format. Scaloperas UTA meg. Okay, now I can go back to my mega window. Where is it? Okay, so I'm just going to find that window, right? So I'm going to open a new file. This is going to be the Scaloperas UTA.meg. I guess click yes. Okay, I'm going to go ahead and close out myself from. Okay, let's go ahead and create a session file. So Scaloperas MDX. Now I've closed everything down I had before. And so I'm going to go ahead and open the Scaloperas UTA.meg. I can go through the same steps. Compute pairwise distances. Scaloperas UTA meg. Yes. Okay, default. And you might notice that the outgroup has much larger numbers, okay, because the outgroup is less related compared to all of these Scaloperas. Scaloperas are more closely related to each other. You want to see the entire matrix. Open up the window a little bit. You can go ahead and Command Shift 4, and you can select the portion that you want to incorporate into your presentation. That's your genetic distance matrix. And now I can go ahead and click up on phylogeny, construct UPGMA tree. Yeah, use the default setting. And there you go. How beautiful is that? Okay, so the one thing that is baffling me right now is why I've got a no branch lengths on the UTA, but everything else looks really great. In fact, what's nice about this, and sometimes this doesn't happen, depends on how successful you are in choosing your outgroup. The outgroup doesn't always come out on the outside, right? And so if UTA came out more closely related to Scaloperas caudas, that would be totally perplexing. I mean, a couple of possibilities there. Maybe UTA is actually secretly a member of the genus Scaloperas. It really was misclassified. Right? Or, more likely, you might have chosen a sequence for these other guys that was not really comparable with everybody else, right? And so there are many possible explanations for why we might have gotten unusual or atypical result. But if you got a result that looked like this, I'd say, hey, you've just gone through a nice exercise of phylogeny reconstruction. These data are coming from different sources. Okay, you've basically done an analysis that nobody else in the world has done before, maybe, okay, potentially. And so this is kind of like, this borders on original research. And yeah, sure, you're not doing any of the grunt work. You're not collecting the samples. You're not preparing the samples for sequencing. You're not doing the sequencing, but you have the data. You paid for the data with your tax dollars. And that's why they're free for you on the NCBI database. Right? And you're using the data to potentially deepen our understanding of the natural universe. Okay? I think that's pretty cool. I mean, you could go through this exercise and actually come up with novel results. And that's what I'd like you to do. Okay. Now, for your presentation, I'd kind of like you to be doing something like maybe a 5 or so minute presentation, no longer than 5 minutes. Okay. Don't go off the deep end, and I don't need you to give me detailed descriptions of what every species is. I mean, I would like you to inform your audience more or less what kind of animals or plants you're running your analysis. You're reconstructing the phylogeny for, but you don't need to give the natural histories of all the species in your group. Okay, you might want to justify why you chose your outgroup as you did. I'll be able to help you out with all these answers, with your justifications. But try to keep it under 5 minutes. Make sure to include, well, a few things, right? Make sure you include the people that are involved in your research. Okay, so all the members of your group. Make sure to identify the species that you are reconstructing the phylogeny for, as well as the outgroup. Okay? You'll want to identify the molecule that you compared. Okay, and that's really important, because if I do this analysis and I come up with this phylogeny reconstruction for these Scalopera species using NADH dehydrogenase, and you do the same species, but use a different gene, maybe use cytochrome oxidase 1. Okay? You could totally do that, right? Maybe somebody here will do exactly what I just said. Do the same species that Ihara did in his demo, but use a different molecule, cytochrome oxidase 1, which is also there in the mitochondrial sequence, right? Okay, can't have everybody doing the same thing, but that's one possibility, right? And so if you were to do a different sequence, you might come up with different results, right? I think anybody that ran NADH dehydrogenase would probably get results that are similar to mine. Somebody that runs a different molecule might get different results. And so the results that you report are definitely molecule dependent. So in order for your results to be replicable, in order for somebody to be able to replicate your experiment, your research, and come up with the same findings, you need to provide the information. And so if you don't include information of what molecule you used, that makes your report result non-replicable, and that's not a good thing. Okay. But as far as your results go, you should include the phylogeny, the cladogram, showing the branch lengths, and you should also use show the pairwise distance matrix. Okay. Now, one of the things I want to share with you here is how to interpret the branch lengths that we're seeing here versus the information that we have in the genetic distance matrix. In fact, these numbers should have a pretty tight correspondence. Okay, so let's take the comparison between Scalopera occidentalis and Scalopera virgatus, right? And so the way this works, we have 3 sections of branches that separate these 3 species. Okay, so you've got a horizontal branch here, you've got a horizontal branch here, and you've got a horizontal branch here leading to occidentalis, right? Now, each of these branches corresponds to a branch length. Okay? And so 0.06, 0.01, 0.05. And the idea here, so we're taking the genetic distance between Occidentalis and Virgatus, and we're parsing it out into the differences that have evolved from this ancestor here to Occidentalis. Okay, the differences that have evolved from this ancestor back here to Virgatus, and the differences that evolved between this ancestor shared between Occidentalis and Virgatus and this ancestor that Occidentalis shares with these other species of Scoloporus, right? And so if you want to look at the overall genetic distance between Occidentalis and Virgatus, you need to add 0.05, 0.01, 0.06. That comes up to 0.12. And that should be more or less pretty close to the genetic distance that's reported over here, right? So if you look at the distance between Scoloporus occidentalis at species 2 here and Scoloporus virgatus, it's showing us 0.11. Not the same as 0.12, but that's completely within the possible rounding error. These numbers shown in the branch lengths are not highly precise. I mean, there could have, rounding error could account for the differences that are there. And it's also not going to be exact, even if we made these numbers more exact, more precise. They're not going to add up perfectly well, but there should be a pretty good, strong correspondence between the branch lengths that we're seeing in the cladogram and the genetic distances that we're seeing in the genetic distance matrix. All right. So I know that's a lot. It's given you quite a bit to think about, but what I'm hoping is that the information that I've given you here will get you on your way to producing a really cool presentation to share with the class. And I will be putting together a short demo on how to use Canvas Studio to make a little video to post into the discussion board. And so your deliverable for this is going to be a short presentation including all the information that I've given you here. I've told you to incorporate into your presentation no longer than 5 minutes, okay? And it should involve everybody in your group. All right. So sorry for the long video, but that is a complete walkthrough. And again, if you have any problems, go ahead and bring those questions to me in class, or you can put them on the board, on the discussion board, or send me an email or something like that. Okay, I'll be happy to help. All right, bye now.